Okay, thank you guys all for being here. Again, I'm Mara Cowan, and in this session, we're going to talk about model systems and exploring GRI disorders through model systems and what they can teach us about GRI and how they can bring us closer to treatments. So to start out, what is a research model? So a research model or a model system is a living system that scientists can use to study how biology works. And so these models can help answer important questions about human health and disease that are really not feasible to study in humans. For ethical reasons and difficulties and challenges, things like screening drugs really can't be done well or fast in humans to find something that might be a good target to go after, like we talked about in today's earlier sessions. And so there are many different types of model organisms that people can use to study something like GRI. But today we're going to have Dr. Howard Sorokin and Dr. Amy Ramsey take you from fins to furs, highlighting some of the amazing work that they both have been doing studying zebrafish and mice as model systems. So model systems are really crucial because they allow researchers to ask questions about what's known as the underlying mechanisms of GRI disorders in a controlled experimental context. And so this allows you to directly link gene variants to things like synaptic, cellular, and system level changes. And so in other words, how do genes affect neural function and ultimately behavior? Oops. And of course, we can learn really important information from human studies. So the things that you fill out, like surveys or samples that you give, like many of you did yesterday, can be used to compare what's different between individuals with GRI disorders to those without a disorder. But to go after the mechanism, what you need to do is you need to break the gene and then see what happens. And to do that, it really depends on a model system. Now, the model system that a scientist decides to use for their research really depends on the question that they're trying to address. But just in general, a good model system tends to be easily modifiable genetically. So this means that we can manipulate the genes, we can turn them on and turn them off, we can break them, we can overexpress them in the system. They need to have a relatively fast reproduction or be easily expandable for the speed of doing the research and doing the science. Ideally, they have a well-characterized genome that'd be similar. What similar means depends on the model system, but sort of similar, at least in the genes that we're interested in studying in that system, to that of humans. They need to be affordable and can be maintained in the lab easily. And then obviously we want it to be ethically and practically accessible. And so I'm just going to go through some brief advantages, and for those of us who are studying these other model systems, please don't take offense. So mice, for example, have a really highly conserved genome. Their brains are much less complex than a human, but they do share some of these key features of humans, huge structures, and they are very well established. They're a well-known model system. It's been studied for many, many years with things like genetic tools and behavioral assays being really commonly accepted. They are a little slower and more expensive than some of the smaller models. They're not ideal for screening drugs, although you can certainly test drugs in them, but doing like high throughput screening is difficult. And it can be difficult to link certain behavioral changes to things that are happening at the individual cellular level. Zebrafish are really advantageous in that they are transparent. You've probably heard a lot about that. I mean, they're see-through, so you can do things that are really cool, like real-time imaging, and they are really good for high-throughput screening. They are a little bit less established as tools, and they do have a simple neural circuits that differ from humans, but they are still a great model system to study neuro in, and they do have somewhat limited behavioral readouts. And then there's also something this is not an actual living animal, but known as iPSC-derived neurons. And so what this means is you can take a cell that's already a cell, something like a skin cell. It's already decided what it's going to be. You can actually reprogram it back into a cell that can become any type of cell, which is called a pluripotent cell. And then you can turn it into the type of cell that you want to study in a dish. So this is something like a neuron. And these are really advantageous because you can get patient-specific neurons to study in a dish. They are limited in their development, but they mimic early human brain development, and they can allow for things like the advancement of personalized medicines. You cannot study behavior in cells in a dish. You could study things like how they signal. So you can certainly do that. But in terms of behavior, how one animal interacts with another animal, for example, you cannot study that in these types of cells. They do have limited maturation and circuit function, especially for 2D, although 3D helps that a little bit. And then they are isolated, so they don't exist in the context of a brain. So the best thing we can do as a research community is really collaborate with others and study different model systems, like in this conference, and inform each other, which would help expedite the pipeline from bench to bedside. So how do model systems help GRI research? So we can do things like uncover gene function. We can knock out or basically turn off a gene in a model system and study the effects of that. You can study human-specific mutations. You can use patient-specific mutations to learn about specific variants in receptor function or downstream behavior. You can test potential treatments through screening. And then importantly, you can also control gene expression in things both like time, so temporally turning it on at specific times, like possibly in adulthood, and space, so in specific cells and populations in the brain. And so I'm just going to briefly touch on this before we dive into some science, but this is the type of work that I did as a PhD student, and I studied another model system called C. elegans, and so that's a transparent nematode or worm, really tiny microscopic clear worms, or as Steve said, the zebrafish of worm, but they develop rapidly, they are self-reproducing, so they don't have to mate, they have a mapped connectome, which means we know every single neuron that connects to every single neuron in the worm, and 90% of synaptic genes are conserved. They also perform stereotyped behaviors, and so I'm just going to talk to you about this one type of behavior called social feeding behavior. What I'm showing you here, this like black circle, is actually a group of worms, and these individual squiggles are individual worms. And so you can see very clearly from this image that social worms are together and solitary worms are spread out on the plate. And so instead of trying to ask questions about how genes control neural circuits and behaviors in the human brain, which consists of 90 billion neurons and hundreds of circuits, each connected by thousands of synapses that control multiple complicated and unknown behaviors, we can use C. elegans with just 300 neurons, in which we can study individual neurons and circuits that are made up of just a handful of synapses in which we know the behavioral output. And I'm really happy to talk to you guys about this in depth, but I'm going to quickly just take you through 3 quick findings from this about glutamate. So what we saw is that in social animals compared to solitary, the solitary animals had slower transport of glutamate. And I can talk about how I did that at another time. Additionally, if we got rid of or knocked out this essential glutamate transporter, so in this no glutamate signaling here, we can see this very clear reduction that I didn't put on the map, but it's very significant, I promise. But very clear and significant reduction in aggregation behavior compared to the controls. But we also saw that if we bring back this glutamate signaling in just 2 neurons of those 300, then we're able to rescue or restore that behavior. And then lastly, of interest to this audience, I did look at which receptors were involved in this. And so here we have these, they're called homologs or orthologs of the human genes. And so the NMRs are the NMDA-like receptors, so like the GRINs, and then the GLURs or the DLRs are like the GRIAs. And so what you can see here from this is that it doesn't seem like the NMRs are involved in this behavior, but you do see a reduction in the social behavior if you have a GLUR1 or GLUR2 mutation. And lastly, I'll just show you what that looks like here. So that's the control being social, the NMR mutant being social, the GLUR1 being less social, and the GLUR2 being less social. And then this is the restoration in those 2 specific individual neurons by bringing back this gene called E4 that's important for this glutamate vesicular transport. And then importantly, this is the type of system that can be used for high throughput screening, so we can screen FDA-approved drugs on this in a very high throughput manner, which just means that we can do things like put worms on individual wells of a 96 well plate, and we can record their behavior and watch them move, like what's shown here. And with this, we can screen about 100 drugs per week, assuming everything goes perfectly, but about 100 drugs per week, which would be really, really fruitful and really helpful to then be able to say, this is a good drug that we could use, and pass it on to Amy, for example, to test in her mice. So thank you guys. And with that, I'm going to introduce Dr. Howard Sorokin, who's going to talk to you guys about the zebrafish work. Thank you.
Okay, so that was a really nice introduction. Thank you. All right, so what I wanted to do today was to talk a little bit more about the zebrafish and why you're hearing so much about it today, and then just show a couple of slides on our work on the role of GRIN genes in early neurogenesis and in developing GRIN models. So many of you have been really kind and open about sharing your stories about how you got here, and so I just wanted to tell a little bit about 30 seconds about my story and how I got here. And so many of you may have children who for reading a book as part of the bedtime ritual. When I was about 3 years old, my older brother worked at a library, and they were throwing out this book called Buffalo's Story of a Toad, so he gave it to me, and that was my go-to book that I had to, be read every night, every single night. I knew it forward and backwards. My parents or my siblings had to read it to me. It's this geeky little story about a tadpole turning into a toad, and I was amazed as an adult when I came back to that book, and I opened it up, and I looked at the second page of that book, and this is what I see. This is a picture of a frog embryo dividing. It's a one-cell frog embryo, 2 cells. This is blastula stage. This is gastrula. It's a very beautiful neural plate. This is exactly what I study now, right? And this really illustrates one of the advantages of zebrafish, is that because of the transparency of the embryos, we can see what's going on in early development and at later stages. Okay, so if we're interested in rare diseases, why zebrafish? Okay, so I just mentioned that the transparency allows us, and I'll show you examples, to see a lot of very interesting biology. And the genetic tools that we have allow us to study those mechanisms. But also, and particularly important in terms of rare diseases and GRI disorders, it really provides an excellent platform for high throughput behaviorally based small molecule screens to identify new molecules that may be therapeutically beneficial. Okay, so many of you have probably seen adult zebrafish. You can go to Petco and buy some. You may have some in a tank at home. The adults are about 2 to 4 centimeters long, an inch and a half. When we talk about zebrafish, we're focusing on a couple of different stages. So very early embryo, so this is a 1-cell embryo here, and it develops very rapidly. So it goes from 1 cell to this very complexly patterned animal in about 24 hours. So we can watch what's happening, we can manipulate it. But most of the work I'll talk to you about today, and you've heard about from others at the meeting, is really on larvae. And so this is what a zebrafish larva looks like. So this is the eye over here, so this is the head, the tail here. And what you can see, one of the things that is really obvious is you can see through this animal, right? And that opens up a lot of biology. The other thing is that these animals are freely swimming and freely behaving. And so we can do some actually fairly complex behavioral assays. This transparency does a lot for us. And so what you're looking at here, this is actually this is a living zebrafish. It's embedded in agarose. It's actually the work of Misha Ahrens. And this, what you're looking at, these are different optical sections through the same animal. Okay, so just going from the top of the head going down, different levels. And this animal happens to contain a transgene, some DNA that's been added for a calcium indicator. Okay, it's a genetically encoded calcium indicator. And you may remember, from all those slides looking at NMDA receptors, right? There's calcium going through the channel, right? This is, when people talk about neurons firing, those are calcium fluxes. And so what this reporter does is it fluxes in response to calcium levels. And so each of these little dots that you see here are different cells that are firing, okay? And we can do this through the whole zebrafish at single cell resolution through the entire brain. And this larva is really in a virtual reality environment. So there's different stimuli being presented and stuff like that. But we can do that in living animals and potentially in GRI models also, in GRI models. The other thing that's really useful is it's a great system for high throughput behavior and high throughput drug screens. What you have here is a 96 well dish, so it's a 12 by 8 array, as was just mentioned, and in each well is a single zebrafish larva at about 6 days old. In this case, these flashes are just to highlight that the fish is moving. Okay, so every flash is just moving somewhere. So what interesting things can a fish swimming in a little well like this do for you? Right, so we can look at its movement. Is it hyperactive? Is it hypoactive? We can also look at seizure susceptibility. So is it having behaviors characteristic of seizures? We can also study learning in this format, habituation and other forms of learning. But what's really useful for the disease-related studies is that we can supply test compounds into the water, okay? And the fish will take them up very easily, okay? And so that enables us in one of these kinds of assays to test 96 different molecules at a time, okay? And so some of you may be familiar with fenfluramine, which is an anti-epileptic that's in common use. It was really only about 10 years ago that it came from a screen like this, okay? Because you're able to screen, dozens, hundreds, thousands, even tens of thousands of molecules, depending on your setup, which is really powerful in terms of identifying potentially therapeutic molecules that could be then switched to other model systems, other preclinical models like rodents and then preclinical studies. So the idea is that the throughput is just much higher, where in rodent models, you're typically testing one compound at a time. And here we can test dozens of compounds at a time. In a single 96 well dish, you can be testing multiple dishes in parallel. Powerful for unbiased drug screens. So one obvious question is, are zebrafish really good if we're interested in complex behaviors, right? Obviously, their brains are smaller. They're not as complex as ours. So I just want to take you through one example from our work. So we've generated very strong loss of function models of 2B. Okay, so this is more of a complete knockout. And we wanted to ask about the behavior of these animals. And so one of the behaviors we looked at are their social interactions. So in this case here, we're testing in this apparatus 10 fish. So each lane is a different fish. And they can't see each other between the lanes, but what they can see is at the end of each lane, and at both ends of the lane, there's a window. And behind that window, we either we leave it empty, or we put a conspecific fish. And we ask the question, how social is this fish? Does it like to swim up here in zone 4, which is by another fish, or does it prefer to be in zone 1 by itself? And so if you do that with a wild type fish over the course of development, so this is looking at the first 4 weeks of development, what you see is that somewhere between the second and third week, these fish begin to develop a social preference that becomes even stronger at 4 weeks post-fertilization. And that's just shown here as a social preference index. If we do the same thing with a GRIN2B knockout fish, what we see is that they fail to develop that same social preference. Okay, so that suggests that obviously, humans are much more complex, right? No question about it. But that some of the underlying brain circuitry that drives social behaviors in humans is regulated by GRIN2B, and the same is true in fish. But we can also use a fish to dig deeper. And so now we took those same fish, and one of the things people have mentioned at the meeting, to talk about the balance of inhibitory and excitatory signaling. And so we've looked at that in these 2B knockout fish. And these animals, we've now crossed in a couple of reporters. One of them is red, and that's marking excitatory neurons. The other one is green, it's marking inhibitory neurons. What you're seeing is a series of optical sections going, again, from the top to the bottom of the fish. So this is part of the optic tectum, the telencephalon. Different brain regions are going to come into focus as we go deeper into the brain. And so there's lots of, as was mentioned, lots of excitatory neurons, fewer inhibitory neurons. As we get into the telencephalon and up here, we'll see a lot more inhibitory neurons in the deep regions of the brain. And so we're able to use this model to compare, to ask the question, what's different in the 2B from the controls, from their wild type siblings? And what we see is when we look at various parameters, that some of these interneurons in these regions here in the ventral or bottom part of the telencephalon, that there's fewer of these interneurons in this zebrafish model. And that was really interesting to us because the regions in zebrafish correspond to things like the amygdala, the striatum, and the septum in humans, which are regions where interneuron deficits in individuals with autism have also been found, or deficits in neurons. So we think that there's this model is showing a lot of both face validity and construct validity to human patients. In addition, these animals provide an opportunity for us to look at things that are difficult to look at and to find novel findings. So one of the things that we've done, and this is actually the work of Christy Priya, who's a graduate student who's at this meeting. She's in Lonnie Woma's lab, and she's looking at the enteric nervous system. And I know from talking to everyone here how big a quality of life issue the GI symptoms are for patients. And so what we're looking at here, and we have what we think is kind of a novel finding about what NMDA receptors do. And so this is a wild type fish, and what's in green here are the cells of the enteric nervous system. Okay. And now if we look in GRIN1 mutants, what we see, there's many more green spots, many fewer green spots rather, or if we treat with an NMDA receptor antagonist, there's many fewer cells in the enteric nervous system. And so we think that a lot of, and this stems from a defect in a cell population called the neural crest. And so we think that actually may be, and we're looking into this further, really strong contributor to what's going on in terms of GI symptoms in terms of GRI patients. Because these are the neurons that are involved in peristalsis and moving things through the GI tract. Okay. The last thing that is very advantageous about zebrafish is the scalability. We can study many disease-associated variants in a living dynamic system. We've taken advantage of we've recently taken advantage of you may have heard of the CRISPR system for gene editing. This is prime editing. It's a variant of CRISPR. And so in our first efforts, we were trying to model 5 different GRIN gene mutations, one in GRIN1, 1 in GRIN2A, and 3 in GRIN2B. These two here, so this one, L81, GRIN2A, L812M, and GRIN2B R696H. This one we haven't been as successful with. Both of these are from CureGRIN families. Okay, and we're interested in understanding both in terms of what's different about these fish on many different levels. We started out doing some behavioral assays. So the first of these models that we developed was this GRIN1A M706V, and we ran the same social deficit, the same social assay, and we see that these animals also display a social deficit. And so some of the stuff that's in progress and future plans is that we're obviously looking at these and the other models, but also assessing seizures. There's a graduate student in Lonnie Woma's lab, Erica Nevitt, who talked a little bit about this on Wednesday in her talk. Can also assess learning, but importantly, we can also test the consequences of or the effects of anti-epileptic drugs that are in use to see which ones might be effective for a particular variant. So that's personalized medicine, something that we would like to get to, and we have the opportunity to do with some of these. Okay, so just to kind of sum this up, the reasons that zebrafish are useful is because first of all, they display complex behaviors. They're obviously simpler than humans, but the behaviors do resemble ours in many ways, including social interactions, learning, and seizures. It's a very tractable system for studying brain pathology and pathogenic mechanisms. We can look at cell fate, we can look at cell movements in very sophisticated ways. As I mentioned, in our 2B knockout fish, we see both deficits in social behaviors and in interneurons that resemble what's observed in human patients. And so, and the variant lines that we're developing, and part of the strategy is that it allows us to look at lots of different variants, right? And try to look at some gain of function and some loss of function and ask questions, does a 2A and a 2B gain of function? Do they look the same or do they look different? And what are the similarities and differences between these? And hopefully be able to assess, efficacy of different kinds of treatments, but also potentially to screen for novel drugs. Okay, and really, as I mentioned, that from zebrafish screens for Dravet syndrome, fenfluramide really came out of that. It really presents a really excellent opportunity to test, novel compounds to alleviate, potentially alleviate different kinds of phenotypes. So here's my zebrafish. And lastly, I just want to acknowledge people in my lab. So the disease model generation has been done by Gina Rizzo, who's right here. All of this work is a collaboration with Lonnie Wilmoth's lab, who's also at Stony Brook University, and Christy Apria and Erica Nebbit were both at this meeting. Christy's the one who's doing the neural crest work. So thank you.
Now we're going to have Dr. Amy Ramsey join us to talk about the mice.
I feel like I misunderstood the assignment here. So, Howard, that was such a good talk. I don't think I'm going to be able to hang in there with you. Okay, so why mice? I want to go into that, but first I want to preface this by saying that all of our research is guided by a lot of ethics, which I really appreciate. And so I wanted to introduce you to this ethical concept that guides what we do. It's called the 3 R's. The first that we need to really think about before we do an experiment. The first is replace. Is there any way that we could do these studies without animals or in what they call a lower order animal? And I mean, that's a very that's a very human-centric idea that other animals are lower than us, right? So how far away is it from the human? As far away as it is lower, and we're at the top of the order, right? So, but that's what they call them. Can we do it in something that's farther away, less sentient, less feeling, less emotions? But really, we do try to do that. Reduce. Can we reduce the number of animals that are used to the minimum amount necessary for good science? Now, it's really bad science to do an experiment on 1 animal. It's actually, you might as well do it on 0 animals, right? So you're wasting that animal's life because you didn't do it in a way that you could actually tell what's happening. So you need to use enough animals that you are sure about your conclusion, but don't do more than that. And then the last R is refine. We need to make sure that everything we do, we're constantly learning about the mouse and trying to find ways to make their lives better, to make them healthier, to make them more comfortable, just in the same way that we learn about dogs and we know, oh, well, maybe we should be feeding our dogs this instead of this, or, oh, dogs don't like this. Let's, like we care about our dogs more and more, and we treat them differently. Same with our mice. As we learn more about them, learn what they need, we try to give them that so that they have a really good life in everything that we do. Okay, I'm just jumping right in. There are things that you learn from animals that you just can't learn from cells. And this slide is from a talk that I gave a couple days ago. And the talk that I'm giving today isn't really heavy on science because I thought, oh, this isn't the science talk. So let me just back up and tell you, one of the things that we learned studying the GRIN knockdown mice. So we always were looking at their brains, but we started talking to parents and realized we need to look at the whole animal the same way that a doctor looks at a whole person. And so we started studying other parts of their body, including digestion and glucose regulation. So we started looking at their pancreas, and wow, I've been working with these mice for 25 years, and I didn't know this, but their pancreatic islets are smaller and fewer, especially in the males. We could never have learned that from, just working with cells, neurons in a dish. So we just can't know how these genes affect every cell of our body working together. Okay, so I wanted to also point out that even though humans and mice are, of course, very, very different, if you think about the whole, animal kingdom, we're actually pretty closely related. So this is a diagram here showing the millions of years between when we had a common ancestor. And we are much more related to mice than we are to other mammals like, dogs and cats and other things that we think of as mammals. So that are maybe even closer than our size, like pigs or bears or something like that, right? We are descended from a similar ancestor, and our genes are closer together than some of these others, like birds and other mammals and so on. So there's a lot of advantages to working with mice. I'm going to talk about those, why we would want to use them. They're small. So a lot of this, Mari, you talked about. And so I'm like, okay, well, yeah, I'll talk about it again. They're incredibly reproductive. So they have a new litter of pups every 21 days. So pretty much the moment that they are like, okay, you're done nursing, I'm having my babies tonight, and then it starts over, they never get a break. And then they get pregnant right after they have their babies, they get pregnant. So they're nursing and pregnant the whole time. It's and it's like clockwork. You're like, when's the next litter due? They just had babies, so in 3 weeks we'll have another litter of babies. That's quite a lot. We have so many tools to change their genes. The other thing that we can do is these mice, they all are identical twins, so they have the exact same genetic background. When we're studying humans, we're so genetically diverse. And then, of course, siblings are related, but they have different ages. And then if they're fraternal twins, then, well, at least they're the same age, but they're still pretty different. And it's only with identical twins that you're like, okay, they're pretty similar, and that's what you can get with mice, is that they all are identical. They've bred brothers and sisters together for like 20 generations until pretty much they're all the same. They're nothing like a wild animal. They're nothing like a wild mouse. So we have domesticated them and chosen the ones that don't bite us when we pick them up and don't run away. So we are not working with wild mice. We've domesticated them. Okay, so we've established a lot of methods to study them. We already know so much. We've been working with these animals for so long. So we're building on a lot of knowledge, and the same with these other model systems. The elegans, the zebrafish. The more we know, the further we can go. They have a similar brain organization to humans. Of course, it's less complex. They're social animals, which is really important if you're trying to understand what makes us social, or what are the things that cause us to be less social. You would want to study this in a system where they are also social. And there's something that's unique about mammals. I mean, our reproduction, lactation, this oxytocin causes some bonding, and there's just some things about being a mammal that are unique to mammals. So I think it's important that we're working with a mammalian, and especially the glutamate receptor sequences are very, very similar. There's very few differences at the amino acid level. Okay, here's the problems. The brain is certainly less complex. The mice don't have language, just vocalizations. One of the most important things that parents care about is communication. and I'm really not sure whether we'll be able to ever address that in mice. Pair bonds are not as strong in mice as they are in humans. So they're social, but their social relationships are different. They have they're very short lifespan of 2 years, so we can't study a lot of aging things. Very different metabolic rate. They metabolize drugs about 10 times faster than us. They need to eat 10 times their body weight. No, I'm sorry. They eat 1 gram of food for, 1 gram of food for every 10 grams of body weight. So they're eating 1/10 of their body every day, not 10 times, 1/10. But still, if you weigh 150 pounds, that means you're eating 15 pounds every day if you're a mouse, right? That's really different. And they have a different, let's hope, they have a different diet. So they're herbivores, and we're omnivores. So, that actually, it's the herbivores that have all the metabolic enzymes to deal with the things that are in plants. So carnivores that don't eat plants, they metabolize drugs differently. That's why we can't give a lot of our medicines to our cats and dogs, because their bodies they don't have the machinery to break it down the way humans do, because we eat plants. Okay, so drugs that work well in mice don't always work well in humans. They're sentient creatures. There's a lot of ethical issues around that. It can be very hard for humans to work with mice too. And animal studies are very expensive. So all of those things we take into account. Really, as much as you can do and avoid using mice, you would want to try to do. But there are just some things that we can only do. So let's think about what is important to patients and can we study it. So the communication is one that is very important, that I really am skeptical that we can study it. However, animals do make vocalizations, and I see that as a fine motor skill. So we do see changes in vocalizations, and I like to think of it that way, that, the way that you shape, your larynx and your mouth, those are fine motor skills. So I think we can study that part of Grin disorder. Parents care about behavior, and I think this means like emotional outbursts, really, when they say that. Intellectual disability. So these are things we can study. Emotions. We can study different aspects of cognition. We can study seizures and be really good about predicting whether something's going to be an anti-seizure drug in humans. We can study, the biology of gastrointestinal physiology. We can study sleep really nicely. Autonomic storms are tough in both humans and in mice. So this is something I think is really important, and we need to get this studied in both humans and in mice. But in theory, we should be able to do it. It would be about continuous monitoring of all of those things like your heart rate, your breathing rate, and things like that. Okay, so Dr. Sirotkin mentioned some things like construct validity, face validity, predictive validity. So these are things that we think about, like it's essentially like how close can we come, or what aspects of a disorder can we try to approach and address in a mouse? And really, I think that the one that you probably care the most about is what we learn in this mouse. Does that predict what we could do in the human as far as treating, treating the disease? But also, we can learn some things from this mouse, and then, we can actually look and see, like, oh, there's a pancreas difference in the mice. Well, let's go see what's going on with blood glucose in the patients, right? I put up this slide just because this is what we're trying to do when we look at behavior. And we're really good, I think humans are really good at domesticating animals and reading their intentions. What are they thinking? What are they doing? We're good at this. So we can look at this dog and say, okay, that dog is alert and excited. This dog is exploring. We're looking at their ears, their tail, the position of their body, and we are getting all of this information, and we would all agree, oh yeah, that dog is upset, don't go near it. We know. We're not so good at mice. But we can say okay, these are the first mice that I made when I was a graduate student. These are the GRI knockdown mice. So these are wild types here, and they're all huddled up. And these are the GRI knockdown mice here. So, I saw them in the cage. I was like, they're not huddling. They're supposed to be huddling. I don't know what they're thinking, but I know they're not doing something right. And so, what I want to do is to be able to know what they're thinking by how their bodies are moving and try and just capture what's going on in their brain. So, I just wanted to finish with some of the tests that we use. This is very similar to what's done in zebrafish. We put them in a new environment and see what they do. And that's like a basic behavior. We're going to get confronted with something new. What do you do? So we have a digital activity monitor where the animal is placed in a square container, and there's something that's measuring where it is at any given time. And you can see that the animal runs around. This is how much they travel in every 5 minutes. The wild type animal runs around, checks it all out, and then goes in a corner and sleeps, because we do this when it should be sleeping. And this is the GRI knockdown. Super hyperactive in this new environment, and really takes a long time to settle down. And in this animal here in red, what we're looking at is a genetic rescue. So this is where we try to improve this gene function, and we could see that, yeah, they're still hyperactive, but they settle down a lot faster. Here's another one that we did, where we are now looking, okay, let's see if this animal can solve this problem. This is a really complicated behavior because they need to be motivated, and they need to figure out how to solve the problem, and they need to figure out what to do when the problem changes. So first we put them in this open box, and we show them, look, there's a door, and you can go into this darker area, which mice generally feel safer in the dark. Then we block up that door, so they have to crawl through this underpass to get to the goal. And then we fill that underpass with bedding, and they have to dig through it. So we're looking at this combination of, do you remember, can you change, and can you solve a new problem? So just with the first door, you can see that the mutants, they have some motivation issues. But then you start to give them the closed door and fill it up with bedding, and we stop the test at 5 minutes. So they really have problems. And the wild type, they are fastest when the door is open. They quickly figure out, okay, yep, I can use this underpass. And then the bedding, of course, they have to dig through it, but they solve that too. And the rescue mice, it was really excited. We could see some aspects were just like wild type, and then in all these others, they were better. So that would be how we would say, okay, we've got something here. What does this mean for humans? Hard to say, we are trying to make a big leap between what we're seeing in mice and what we're seeing in humans. But what we know is that it's closer to wild type, and they're doing some behaviors that require some types of cognition. Here's some other behavioral tests that we do. So here's the locomotor box. We can look at anxiety by looking how much time they spend out in the open versus in an area where there are close walls. We actually see that the animals don't like being closed in, and they spend more time out in the open. So they don't like walls really close to them. But then if you put them in a chamber where the walls are like this, they spend all their time there. So it's not so much that they aren't afraid, but they're afraid of something different. They really don't like close spaces. Maybe they don't like their whiskers touching on the walls, but we can see these kinds of changes. And We can do things like measuring their seizures. So we would just pick them up and hold them, set them down and see if they have a seizure. And they develop seizures. If you do this once a week, around week 7, you start really seeing everyone having seizures, and you can score them based on how severe their seizure is, and you can see that over time, these animals start developing seizures. So I think, okay, I'm very close to the end. I can see I'm out of time. Okay, so this is this vocalization I was talking about. We can't hear these because the mice are quiet as mice, but if you listen with a microphone, you can hear they're making sounds in the ultrasonic range beyond what our ears can hear. And they actually have these different patterns. So they're making different types of sounds. And then we can study in the mice, are they making the same number of sounds? Are they as complex as the wild types and things like this? And this is something that's done very early on. So we can look at kind of their postnatal development and see that the mutants are making fewer of these vocalizations. Okay, so I think that's, the end. And I thought I would have more time for discussion, but I'll just leave it there and we can all discuss. Okay.
Anyone have questions for our speakers? Or we could discuss, which would be an option.
I mean, I would be happy to hear what people think about the utility of mouse models and, whether you think, because there was this thing from NIH just coming down that they're not going to fund grants that are fully animal research. So that makes all of us raise our eyebrows and we say, what does that mean? And you really think twice, like, can I do this without animals? We're supposed to be thinking that all the time. Can I do this without animals? I don't think I can. But I mean, maybe, should we just be doing this on, what do you think? I mean, what do you think?
Yeah, I mean, I think one of the things, everyone's interested in translational medicine, right? Because we want cures and we want to help people, but I think you have to translate from something, right? And so you have to, learn basic principles in organisms that you can then translate into the clinic. Like the drug screens we just talked about in C. elegans or zebrafish. You can't do that in humans. We see how difficult from the talks earlier today it is to screen, one drug in humans to figure out if it works. Whereas in the animal systems, you can screen many drugs. So I think there's definitely a place.
Yeah, I can only indeed confirm this. Sorry, I'm from the Netherlands, and in the Netherlands, my lab also uses a lot of mouse models indeed to study, in our case, chem kinase, which is very closely linked, of course, to the GRIN field. And then the Netherlands wants to be the first country who's going to be animal experiment free. So I don't know how we're going to do it in the future, but we are facing exactly the same struggles. So indeed, I think that especially also patient advocacy groups can really also make a voice against this, because I think we really need it for the next period of time still, at least for the development of drugs and development of treatments. But I had a question indeed to you, Dr. Ramsey, because you mentioned indeed that of course, we all use inbred strains. And the nice thing of inbred strains is that you can really reduce the number of animals that you need. But thereby we sometimes miss the variation that is seen in patient groups, probably caused by modifier genes. How much do you think that in the GRIN field, modifier genes might play a role also sometimes in the clinical variation seen even between patients carrying basically the same variants?
That is such a good question. And I sometimes wonder how much of this, so a big problem is that a lot of money and time is spent finding drugs that work in mice, and then they don't work in the clinical trial. And I wonder how much of that is, when we're studying mice, we're studying an unnatural animal in the sense that they all have the same genetic background. And And yes, they are able to survive because they have fixed variants that allow, in every other gene, it's a very unnatural system. So that could be part of it. Having said that, so we have bred the knockdown mutation into Black 6 background, DBA background, and 129 background. So we did the pure, and the ones that we do all of our experiments in are F1s between Black 6 and 129. Why did we do that? Well, the mutation, you can get minus animals on a 129 background. So that background will tolerate that severe mutation. But if you put that on a Black 6, 80% of the animals will die. If you put it on DBA, I can't remember whether it was more, or it might be that none of them survived. So the genetic background is very important to whether these animals even survive. So much less, how severe is their phenotypes. And we are so much trying to just reduce the number of animals, try to do the minimal number of experiments, that the idea of putting it on other genetic backgrounds is like, that's just another step of complexity that we don't do, but maybe that's the wrong thing to do. I don't know.
Yeah, I had a question. So what are the steps to get to clinical trials usually? So after the mice model, like if you rescued the knockout one, and then what are the steps? Like, does that go to humans or like what?
Right, so the next step after mouse is that you would want to have it replicated in a lab that is a good lab practices lab. So it's a kind of a certified company lab. They make sure that they see exactly the same thing that you do. So you get it replicated by another set of hands. They don't have any vested interest in it. And everything is done to a certain standard, right? And then you also have to do, you would have to have your drug or whatever, your treatment made in a GLP facility, a good manufacturing process facility, a GMP facility. So then you get your drug, and then you do toxicity studies on it. So that is done in a rodent model and in a non-rodent model, which is usually a dog. So all of this needs to be done. They look at every single part of that animal and make sure there's nothing that would raise a flag that this is going to be problematic. And then you put together this package with all of this data showing that it works in this other lab, that it's not toxic in rodents, it's not toxic in dogs, and then that gets reviewed, and then they'll say, okay, now we'll let you put it in a human. So, there's a lot to it.
You said drug, but this is the same for gene therapy as well? So you'll do the, okay.
Yeah, so I was just going to add that there's a way that people a little bit, which is like for the zebrafish screen that came up with, fenfluramine as an anti-epileptic, is that was already an FDA-approved drug for something else, right? So, and there's libraries of approved compounds, and people will screen them looking for novel effects, and then the bar is much lower because it's already a compound that's been through FDA approval, and people, physicians can prescribe it off-label.
Yes. Last question. Dr. Ramsey, can you tell me all together how much one mice cost? I know you tell me 10,000, but
Okay, yeah,
all
so we were
together with
talking about,
shipping.
right, we were talking about how much does it cost to make a mouse? And I said, well, making the mouse is not that much. It can be 10 to 20,000 where I am. But in like Europe, I think they pay a lot more to have it made. Okay, great, wonderful, good. But then it's the housing that costs a lot. So my mouse bills are about $10,000 a month, and yeah, a month. So that's $120,000 a year, that I need to have the funding to run. And I have about three I have 3 big mice that I study, the knockdown, the Q536R, and the Y647S. So that's so $30,000 or I'm sorry, $40,000 a year, my mental math is really bad, per line. That is what it is. So that's why, it's not like we can just make them endlessly, And you want to tell them how much it is for zebrafish?
Just because you're asking. It's much harder to calculate, but it's much less. And the difference is because there's a different economy of scale in that for mice, there's a lot of upkeep. You have to change the cages, you have to feed them and do things. So it's very labor intensive. For us, once we have the fish facility and once we have this infrastructure and a technician doing it, adding another strain is a negligible cost. So we, in my fish facility, like my lab, we probably have, 60 or 80 different strains of fish. Okay, and so it's a different scale that we can work on because there isn't that per fish cost that there is for mice.
And worms reproduce by themselves, just saying. You can just put them on your desk, they grow on your desk. They reproduce constantly, it's $7 for a plate. Thank you guys so much for being here, we do have to move on, thank you.